वेलकम टू ईजीपीजी स्टडी। लखनऊ यूनिवर्सिटी के B.sc एग्रीकल्चर थर्ड सेमेस्टर सेकेंड ईयर की प्रैक्टिकल फाइल एग्रीकल्चरल माइक्रोबायोलॉजी। So here is the experiment first. So let's begin. Introduction to microbiology, laboratory and its equipment. First point, hot air oven for sterilization. It is used for sterilization of glassware such as test tube, pipettas and petri dishes. Here you here you can see the pictures of these glasswares. और इन ग्लासवेयर को स्टरलाइज हम 180 डिग्री सेंटीग्रेड पे 3 आवर्स के लिए करते हैं और and an oven has a thermostat control using which the required constant temperature can be obtained by trial and error. After loading the glassware, the door is closed and oven switched on. The required टेम्परेचर सेट करने के बाद जो भी रिक्वायर्ड टेम्परेचर सेट करने के बाद हम ओवन को ओपन करते हैं आफ्टर ओनली आफ्टर इट्स टेम्परेचर कम्स नियर टू रूम टेम्परेचर। हम ओवन को तभी ओपन करते हैं जब इसका टेम्परेचर रूम टेम्परेचर के आसपास होता है। अदरवाइज इफ द डोर इज ओपन तो जो ओवन के इनसाइड में जो अभी भी जो टेम्परेचर हॉट है तो उससे क्या होगा, जो कोल्ड एयर उसमें पहुंच जाएगी और ग्लासवेयर क्रैक हो जाएंगे। सो इट्स रिमेंबर द ओवन इज ओपन only after its temperature comes near to room temperature. You can see the picture of hot air ovens. Second point, drying oven. They are dried inside the drying oven at 100 degrees centigrade. Still the glassware dry up completely. Third point is autoclave. Autoclaves are used to treat soil and sterilize nutrient media for plant tissues culture, and also uses the steam under pressure to kill microorganisms and spores. It is used not only to slice liquid substance such as prepared media, saline diluents, uh, but also to sterilize glassware. मीन्स मीडिया और स्पोर्स और माइक्रो ऑर्गेनिज को ही नहीं स्टरलाइज करती है बट ऑल्सो जो इक्विपमेंट यूज होते हैं, ग्लास वेयर यूज होते हैं, उनको भी स्टरलाइज करती है। टेम्परेचर एंड प्रेशर इनसाइड गोज ऑन इंक्रीजिंग, then the pressure increase is observed on the pressure dial. Usually sterilization is done at 121 degrees centigrade. You can see the picture of autoclave. इनर चैम्बर में जो हॉट जैकेट होता है वो हीट करता है मटेरियल्स को जिससे कि वो प्रॉपर स्टरलाइज होता है, और जो मटेरियल्स में जो कंडेंसेशन ऑफ स्टीम हो जाता है, स्टीम का कंडेंसेशन हो जाता है, उसको भी प्रिवेंट करता है। द स्टीम अंडर प्रेशर इन द इनर चैम्बर रीचेस टेम्परेचर इन एक्सेस ऑफ 100 degree centigrade, which can sterilize the material kept inside. Fourth point is microbiology incubator. So incubator एक डिवाइस होता है is used to grow and maintain micrological cultures or cell cultures, temperature, humidity and other conditions such as carbon dioxide, oxygen content, जिससे कि जो माइक्रोब्स है उसकी प्रॉपर ग्रोथ हो पाए, हैचिंग और रिप्रोडक्शन. It is obtained in the laboratory by growing them at suitable temperature. The incubator has a thermostat. Thermostat होता है which maintains a constant temperature set according to requirement. The incubator automatically maintain it accordingly. Moisture is supplied by placing a beaker of water in the incubator during the growth period. Means इसमें temperature, humidity और other environment condition को रेगुलेट किया जाता है जिससे कि एक प्रॉपर ग्रोथ, हैचिंग और रिप्रोडक्शन हो सके. सो हियर इज द पिक्चर ऑफ माइक्रोलॉजिकल इंक्यूबेटर. बीओडी इंक्यूबेटर इज बायोलॉजिकल ऑक्सीजन डिमांड इंक्यूबेटर. ये स्पेशलाइज्ड लिबर्टी इक्विपमेंट है जिससे कि हम वाटर में जो माइक्रो ऑर्गेनिम्स हैं वो कितने अमाउंट में ऑक्सीजन को कंज्यूम कर रहे हैं उसको हम मेजर कर सकते हैं. और मेन पॉइंट इज द And BOD incubators are also known as low temperature incubators or refrigerators incubators because they can maintain a temperature range between 15 degrees Celsius to as low as 2 3 degrees Celsius. Is used for incubator in such cases, and these are the equipment and they are the equipped with heating and cooling system to create a stable thermal environment. So this is the image of BOD incubator. You can see you can see point 6 rich refrigerator is serves as a respiratory for thermolabile chemical solution, antibiotic serums, and biochemical reagents at cooler temperature and even it at sub-zero temperature. मीन्स इसमें यही है कि हम इसमें केमिकल्स, एंटीबायोटिक्स, सीरम्स, सैंपल्स, बायोकेमिकल रीजेंट्स ये सब ये सब इसमें स्टोर कर सकते हैं एट कूलर टेम्परेचर एंड इवन एट सब जीरो टेम्परेचर. पॉइंट 7 डीप फ्रिज इट इज यूज टू स्टोर मीन्स इसमें हम कुछ केमिकल्स और सैंपल्स को स्टोर कर सकते हैं. 14 pH meter. A pH meter is an instrument for determining the pH of liquid, samples, and buffers. This instrument measures acidity, alkalinity of a solution, and it ranges from 1 to 14. Usually, buffers of pH 4, 7, 9.2 are available commercially. The electrode is dipped into the buffer. Uh, the tip has both pH and temp sensors. Hot plate. Hot plate is used to heat chemicals and reagents. And hot plate hot plate is made of iron plate, which gets heated by an electric heating element from below. The required degree of heating is obtained by a regulator. 16. Shaking water bath. A water bath consists of container containing water, which is heated by electric heating animals. It is used for cultivation of bacteria in both medium at a specific temperature. इसका यूज हम बैक्टीरिया को कल्टीवेट करने के लिए यूज करते हैं इन बोथ मीडियम एट अ एक स्पेसिफिक टेम्परेचर पे. Alecq colony counter. Enumeration. In enumeration of bacteria in samples, it is assumed that a single bacterium given rise to a single visible colony. And this instrument that is commonly used to count colonies of bacteria or other microorganisms on a plate containing a gelled growth medium. Point 18. नहीं. And this is the image of colony counter. Point 18. Electronic colony counter. It has two types. Handheld electronic colony counter. Second, tabletop electronic colony counter. 9.19 point magnetic stirrer. It is used to dissolve such substances easily and quickly. A small Teflon coated magnet called stirring bar. And this device is used to make a stir bar immerse in a liquid quickly spin or stirring or mixing solution. मैग्नेटिक मैग्नेटिक स्टेयर का यूज हम क्विकली कोई सॉल्यूशन को मिक्स करने के लिए यूज करते हैं. देन पॉइंट नंबर 20 इज सोनिकेटर. Sonicator. It is used to rupture cells during high frequency waves, means ultrasonic sound waves. Point number 21 is vortex mixture. Vortex mixture. It has a rotor whose speed can be controlled on the tip of the rotor. It's a foam rubber top. It is particularly helpful during serial dilution in enumeration of bacteria. And enumeration means counting the bacteria one by one. And this is the image of vortex mixture. Point number 22. Laminar flow chamber. Laminar flow chamber. Dust particles floating in the air harbor microbes, and an UV light fitted inside the chamber sterilizes the chamber before operation. Airflow to protect lab samples and equipment from contamination. And, and this is the image of laminar. Electronic cell counter. Electronic cell counter. It is used to directly count number of bacteria in a given liquid sample. An example of electronic cell counter is the counter. And this is the image of electronic cell counter. Point 24. Membrane filtration apparatus. Point number 24. Membrane filtration apparatus. Certain substances like urea disintegrate and lose their original properties sterilized by. Point number 25. Spectrophotometer means a beam of light of particular wavelength is passed through the test soil and the amount of light absorbed in the measured electronically. Electrical devices. A, a fluctuation of electric voltage in the laboratory is one of the most important reasons, which reduces the longevity of the equipment and sometimes damages them. Thank you for watching my video. I hope आपको वीडियो समझ में आया होगा. So please like, subscribe and share to my channel.